The human body has millions of electrochemical signals that are sent every single millisecond. These signals are sent through what is known as the nervous system, which can be broken up into two parts. The central nervous system, pictured here in pink, consists of the brain and spinal cord, while the peripheral nervous system, pictured here in purple, is of all the neurons or specialized nerve cells that are not in either of those two locations. These neurons are supported and protected by neuroglia, which are present in both parts of the nervous system. Neurons can be broken down into three pieces, which are the soma, pictured here in blue, the dendrites, pictured here in green, and the axon, pictured here in red. The axon is the most important part of the neuron when it comes to an action potential due to the fact that it is an impulse-generating and conducting region. Action potential is the charge that carries impulses along the axon through voltage-gated sodium ion channels, pictured here in purple, which causes the cell to depolarize, and voltage-gated potassium ion channels, which causes the cell to become repolarized. Without these channels, there would be a large membrane resistance due to the fact that the cell is trying to maintain the balance of charges and ions between intercellular and extracellular fluid. In a continuous conduction, the action potential starts at the beginning of the membrane. For a small amount of time, the action potential peaks and becomes positive instead of a negative in charge, which is also known as depolarization. As a result, the localized current, pictured here as orange circle, forms and proceeds to take sodium ions into the intercellular fluid and out of the cell. This current then spreads out in all directions and depolarizes adjacent membranes. Continuing in a chain reaction, the action potential moves along the axon away from the starting site. Since this has been repeated time and time again in many steps, it occurs at the speed of about 1 meter per second or 2 miles per hour. In saltatory conduction, the action potential starts at the beginning of the membrane, but with a small difference. The small difference is the presence of myelin, which is this yellow dot, which serves as a way to protect and insulate the axon in order to speed up the transmission of nerve impulses. There are specialized cells that help produce the myelin sheath that cover the axon, which in the central nervous system are oligodendrocytes, and the peripheral nervous system are Schwann cells. Due to the myelin, the localized current brings the oxalemma to the next node of Ranvier, or a gap in the myelin sheath. From here, the action potential is developed at node 2, and then carried to node 3 by producing a graded depolarization to bring the oxalemma to the threshold again. By utilizing myelin, the transmission speed was able to be increased by around 150 times the unmyelinated speed would have been. These axons and impulses that travel along them have many different measurements that are used on them. One of them is the length constant, which measures the amount of ions that leak out through the open channels during the action potential process. It has been found that large myelinated axons will have a longer length constant than thinner unmyelinated axons, due to the fact that graded potentials can travel over longer distances in long myelinated axons before dying out. Another measurement that is commonly used is conduction velocity, or more commonly known as the speed of the impulse. By knowing that myelin is a great insulator, it can also be concluded that saltatory conduction is faster than a continuous conduction when it comes to calculating the velocity of the impulse itself.